Okay, so thank you for all to being here. I will be talking about the Regina model. It is applied for virtual cell modeling. It was originally, the idea came from a challenge where it was necessary to predict how certain gene perturbations knockout will affect a cell state. I tried to keep it short. So the problem was in the computational science sense that we are having unpaired distributional problems. We are having cells that are the control cells, and we are having cells that are perturbed. And if we want to measure a certain cell, we need to lyse it, and it means that the cell will be dead. So we have only one measurement on a given cell. We cannot measure a cell without perturbation and after perturbation. So this is the unpaired distribution problem. And what we want to do is to find that mapping function that maps a cell to a perturbed state after applying a perturbation. There was many previous methods to address this issue. Some of them used additive latents. They assume that perturbations in the latent space are vectors. They can be added, subtracted linearly. And it's true sometimes, but there are certain cases where this linearity is not true. For example, epistasis. Other approaches where they use the existing knowledge, external knowledge graphs, like the gears, where they use the geo database. They assume that these databases are complete, and they also, to their results, they used only the highest variable genes and pruned the other ones. And our method is coming here, which is a fully data-driven method. We are not discarding any genes which are not that highly variable, and we are not using any existing knowledge graphs or something like that. What we are doing is we are taking the single cell sequencing results, the gene expression profiles in a control state, and then after a perturbation is applied, it's also a measured one, and then we are feeding it into our model, and I will later discuss it. And our model will give a prediction, and we compare it with the So our model consists of two phases. The phase one is where the latent space regularization is happening. It is necessary because otherwise the training might be, I don't know, not that stable. So we need to regularize the latent space. The input are the raw gene expression vectors. It can be the perturbed ones or the control ones. It doesn't really matter now. It is fed into a transformer encoder to capture the global dependencies. It is important to know that tokenization is needed because those are very big vectors. And then it represented in the latent space. And then in the latent space, classification is happening. It is because to regularize that space, it is for, some kind of biologically meaningful clusters are appearing, and loss is applied here to minimize the distance between the biologically related stuffs. And then, of course, when we want to get the result, it is fed into a decoder, and then we are getting gene expression results. But in the phase one, no perturbation was applied. It is just setting the scene. And now in the phase two, which is a latent Cycle-GAN, we are having the gene expression vectors, and we are put it into the encoder, which weights are frozen from the phase 1. And then we map the control cells to the Pert state, and we can map it forward. And if everything goes well, we can get in the same place. And it is fed into the discriminator, where the discriminator role is to train until it cannot differentiate between it's coming from a real perturbation or a predicted one. And a big advantage for this is that we can apply it for perturbation that was not present in the training data sets, because we introduced that mechanism where we are having the latent representation of a control cell, the gene expression vector of the normal control measurement. Then we fake it meaning that we are masking where the perturbation is present, not zeroing out because it might have some biological meaning, but for example, giving a negative minus value, and fed it into the encoder, and then we subtract it from the original control representation. And therefore we can generalize it because we do it for the seen and the unseen perturbations. And one thing to note that we are doing this at the regularized space, because otherwise it would be very unfeasible, because we are dealing with more than 20,000 genes times. Yeah, I thought that it was masked out because it was in the previous slide. This is just a recap how the prompting mechanism is happening. It's a synthetic signal. As I mentioned, it is necessary for unseen perturbations. And we evaluated our model, comparing with the other existing models. There was the local precision matrix and the global distribution matrix. For example, for the local, we use the Fiam, which is for the local point-wise precision. It is for when predicting the specific individual genes expression, and this is a mean function. And it can be tricky because a model can learn predict a certain mean, but the distribution's shape is completely different. So this is why it's necessary to use a global metric like MMD or Wasserstein distance. It is to capture how two is in relationship with each other. And if they in a closer relationship, it's better because they are closer. And with the Pearson, the higher the number is, the better, because it measures the linear correlation. And we can see that here, for example, the Adamson data set, our model in the local metrics are beating the others, even when took only the 20 most highly variable genes, which, for example, the gears might, we expect that it will perform better because it is for this, but still ours is the better. And when we are taking the global distribution metrics, we are still achieved comparable results. Yes. And yeah, a few words about the data sets. Our model was having the best results at the replugged data set, which is a genome-wide data set. It consisted of lots of genes, like almost 9,000 genes, and lots of cells, and it was very diverse, like not just immune cells, something like this. The others for like combinatorial focused, or the Dixit was a very small data set. It just showed the limits of our models. And here we can see some results. It is on the Regularized dataset. We can see that the Pearson correlation is really comparable with the state-of-the-art gears model, or in the Wasserstein distance, our results are actually better than the gears, which is the, as I mentioned, the state-of-the-art model. So these are really good results, I would say. And we also did an ablation study, meaning that we are skipped certain parts of the model to see its effects. The most important part is the latent classifier. Without it, there was a severe manifold collapse. The Pearson correlation went down dramatically, but also if we omitted the center loss, we had very bad results, like the Pearson went down. And the latent prompting is also a very crucial part of the model. If you are replacing it with fixed embeddings, like with the additive linear vectors, the results are getting worse. So the conclusion is that this model performs very well if the training dataset is diverse enough and having enough data points. The diversity is really important. If we are having only small data sets which are not diverse enough, then the other methods like the gears, of course, outperforms because it cheats with external knowledge graphs. So this model strength is that it is fully data-driven and balances between the point-wise local accuracy and global distribution metrics. And thank you for everyone.
So we have still time for questions.
Can I ask an annoying question, maybe?
What question?
An annoying question.
Yeah, of course.
So you mentioned that there was some tokenization step for like basically turning your DNA expression into some kind of like tokens. Like what choices were made for like the tokenization here? Because I can imagine if you just have normal like DNA, you only have like very limited number of tokens. Like have larger chunks of the DNA that you basically compress into like a single token, or like
it was a very naive approach. I mean, there was the sequence, and we chunked it sequentially. So yeah.
Okay.
Hi, very nice talk. I have a question on this comparison you do for Pearson and different Wasserstein and other models, like, I mean, MMD, yes, this one. So what is your reference? Like how do you know which one, like, how do you know which one is close enough?
What do you mean by close enough?
Maybe I missed, like, I mean, to the actual one, because you're trying to generate.
So that we compared it to the original model's performance. This was the baseline that we compared.
Which was the original one?
I had our model with all of the parts applied included. We compared when we omitted those parts.
Okay, so because the values for the Regina is not mentioned, so I was just wondering like which one you...
You mean the data set? This figure is for the, it was on the Regularized data set.
I see. No, because here there are like four different models are mentioned, and so I was wondering, I mean, what is your conclusion? So which one you think?
The conclusion is that it's good not to left out any part of it, because all is necessary.
I see. Okay, thanks.